Okay, welcome to the lab for Chapter 11 on survival analysis. In this lab, we study outcomes which are times to event. So, for example, for cancer patients, the event is typically death from cancer. But for other events, for example, we have a publication data set, and the event there is when the document's actually published. And so there's specialized methods for modeling such data. And Jonathan's going to lead us off on this lab.
Okay, thanks, Trevor. Yes, so as Trevor mentioned, what the specialized methods are really, a slightly different loss function. So we're going to see methods that look like a regression methods. That's the Cox proportional hazards model, but it's not a squared error loss and it's not a classification loss. It's a special loss for time data.
And what's something that's interesting with these kinds of data is because you track people or events or observations, and some of them don't reach the event or they leave the study early, so they get censored. So censoring is something that one has to deal with these kinds of data.
Yes, and this is baked into the loss, as you would have seen in the chapter. Okay, so as usual for a lab, we start off with our imports of sort of known packages, and these should be pretty familiar by now. And there are some specific packages for survival analysis that we'll use, and that's in this Lifelines package. So the topics we're going to talk about today are fitting the Kaplan-Meier curves and fitting Cox's proportional hazards model, as well as some test statistics. The log rank test you would have seen in the chapter, and we'll also mention a multivariate log rank test. In the call center data, we simulate some time, so we wrote a little utility function here. If you specify the hazard function and covariates, how to draw time from that distribution. Okay, so our first data set is the brain cancer data set. And we're going to start off by fitting some simple Kaplan-Meier curves to the data. So there are several features in here for each patient. One of them is the diagnosis, and I think that's the cancer type. There's the subject sex. There's this Ki is called the Karnofsky index, a measure developed by physicians to measure somehow the severity of the tumor. GGV is tumor volume, and then there's these key variables here, status and time. And these are the variables that over and over again you'll see in survival analysis. Time is the time recorded for that subject. And status, this is that censoring variable that Trevor mentioned. So if an event occurs, typically status will be 1. So if in this study we have a patient and if the outcome is death, the patient died, then their status will be 1. But if they were still alive at the end of the study, well, then their status would be 0, but we would have known they survived at least to this time recorded in the experiment. Okay, so there are 88 patients here, and let's start off by looking at a simple Kaplan-Meier curve. Okay, so the Kaplan-Meier curve, it looks somewhat like a scikit-learn estimator in that you first instantiate it and then you call fit on it. We'd have to look at the package to actually know if it is a scikit-learn estimator. The Cox proportional hazards model is much closer to a scikit-learn estimator. The Kaplan-Meier curve is really just an estimate of the survival function or the distribution function of the time data, and it takes two arguments. One is the outcome y, and the other is the status outcome. And actually, just looking at the signature, I can tell it's not a scikit-learn estimator, because for those, this would be a set of features. The first argument, that would be x, and this would be y, but that's not how the signature is. Okay, so it has a plot method, and we're going to take a look at it. Okay, so this is what the Kaplan-Meier curve looks like, and it plots here. This is the survival function, S of t, and this is the probability, or this is the estimated probability that a time is bigger than t. Okay, and that's actually very thin pencil. I'll have to fix that.
So that's 1 minus the distribution function.
Yes, let's put that in
You
there.
can make the pen thicker, Jonathan.
Yes, let's see how that is. And that's, well, slightly thicker. 1 minus the distribution function f of t.
Yeah.
Okay, and what do we see here? We get a point estimate of the survival function, and we have error bars here. And one thing we note is that as we get to larger and larger times, we get wider and wider error bars. And that's because, well, the way this estimate is produced, it's sort of, in effect, looking at something like the number of people who have survived past 50 here, roughly, and uses that to compute the variance estimate. But there are just fewer who survived to 50 than who survived to 10. Okay. So often we'll have some categorical variable and that might be of interest, and we'd like to compare survival between the two different levels of the category. So here we're going to do a sort of stratified plot or plot of the Kaplan-Meier curve within both sexes. For this data, we don't have any reason to believe that sex is an important predictor, but we're just showing you how to use a predictor to produce a plot to compare the two estimates.
It looks pretty important because it looks like males die much more quickly than females.
Yes, on this data. Okay.
Yeah.
Yes. So it may be important here, but I don't know. I don't know enough about the source of this data to know whether this was an interesting category. But that's a good observation. Though I guess if we should note probably that the error bars are a little bit overlapping, but looks like there might be some difference. Maybe a bigger sample size would elucidate this. Okay, so all we're doing here is we're just using this group by of the data frame, and that gives us individual data frames that we can use to fit a Kaplan-Meier curve, and we're just plotting those. Okay, one thing I've done here is I've collected the data frame here in this dictionary by sex, because we're going to use that in a few cells here. But this isn't used in making this plot. Just as we're breaking the data into the two groups, I'm going to collect this because we're going to do a test to see whether there's a difference in survival between male and female.
That'll settle it.
Yes, let's see what the p-value is. Okay, so for a categorical variable with two levels, a common test used to test whether there's a difference in survival is the log rank test. And the log rank test basically takes the information needed to fit two Kaplan-Meier survival curves, and that is for the one group, say the male, it takes the time and the status, and the other group, the female, it takes its time and its status. And it gives you some overall test to test whether there's any difference in survival. It's not specific at any particular time, but somehow averaged over all of the time points. And the way this test looks, the way the test is constructed, which you might have read about in the chapter, is that it sort of compares survival, makes a little 2x2 survival at every time point and sort of pools these in some fashion. Okay, so the log rank test, these are the results.
Oh, okay. Good.
The p-value is 23%. Okay, so
I'm saying good for the males.
Yes, yeah, I guess we're okay here. So if this p-value is really small, that would be, strong evidence of a difference between survival in male and female. It doesn't seem to be in the.
Not that you'd want any of these cancers, but.
No, yeah. Okay, so that if we have one categorical variable, the log rank test can be used to compare two groups. But often we have other covariates, and for that we need some kind of regression model. That's this Cox proportional hazards model is the sort of default regression model for time to event data. And it's fit a lot like our other models, except the response again is there's an event time and an event status. Okay, so let's take...
In this first model, we just look at sex, the same one we used with the Kaplan-Meier test.
That's right. This is just a simple model,
yeah.
But later we'll have other features.
Right.
Okay.
And look at, and notice the p-value is exactly the same as in the Kaplan-Meier test.
0.23. That's interesting. I wonder if that's related. Actually, this is a well-known fact. When you have a Cox regression model with a single binary categorical variable, The test. Actually, this is the walled test. It may not be exactly the same one. I think it's called the score test, exactly agrees with the log rank test.
Yeah.
But the walled test, the score test, and the likelihood ratio test, you would learn in some other class, are often very similar. We're not going to dwell on those here, but we're not surprised that this is 0.23. So you can think of this Cox regression model as somehow a generalization of the log rank test. If you're to test for, we might include something like the Karnofsky index, and there would still be a p-value for sex allowing for the effect of the Karnofsky index. That would be different than the log rank test, but so in that sense, the Cox model kind of generalizes the log rank test. Okay, let's just take a quick look at how this is fit. We make a design matrix. It's a regression model, so we use our model spec that we've done before. And so when we call the fit function on the Cox proportional hazard model object, we give it a data frame, and we have to indicate here 2 columns. These are the 2 variables that specify the outcome, right? This is the time variable, and this is whether the observation is censored or not. So this looks like a scikit-learn estimator, except scikit-learn estimator would just have x and y. This again has a little argument. That's because we need 2 columns to specify the response. But other than that, it looks kind of similar. Okay, so we can also actually directly compute the log likelihood ratio test, and that's another test similar to the score test and similar to that walled test, and its p-value is also 0.23. That's related to the fact that the score, the likelihood, and the walled test are often kind of similar.
The score test and the log rank are exactly the same. Says so there.
Yes, this one is not the score test, though. That's what I meant. The score test is the yes.
Right.
Yes. So this class is not all about these different kinds of tests, but it's you'll hopefully move on to do some more statistics, and seeing some of these facts is helpful. And this is just a numerical example of that. Okay. So we're going to fit, a more complicated regression model, just because there might be other there are other features besides just the sex of the patient that are likely important for survival, and we'd like to understand how they affect survival. So it turns out that one of the diagnosis values is missing. So we have an incomplete data set. We're going to remove that with the drop NA method, and then we're going to make a design matrix as we've done for regression before, and then the rest of this is similar to this, is just the same as the cell above, except now instead of having just sex, we have all these other variables. And you can see this diagnosis group. This is the type of cancer, and you can compare. So let's just pause for a second to think about what the sign of these coefficients mean. So the way the coefficients should be interpreted are, the simplest. Let's just write out what the hazard rate looks like for if I just had a single predictor. Very, roughly speaking, the hazard rate for. Let's make a bigger pencil. I guess I can do that. Is that right? Yes. Hazard rate T and X is something like some baseline hazard, right? And e to the beta times x. That's very poor handwriting, but that's not just because I'm writing on a tablet. I have poor handwriting. And what is this hazard rate? This is sort of some instantaneous measurement of how quickly you're going to fail. So if beta times x is positive, that means you're more likely to fail at that time, whether if it's negative, you're less likely to fail. So if we have a negative coefficient like this Karnofsky index here, Ki has a negative coefficient, that means if I have a high positive value of Karnofsky index, then the beta corresponding to that will be negative. And so all other things being equal, having a higher Karnofsky index is sort of associated with longer survival. Okay, so you can use that. Similarly, we can see that maybe meningioma has perhaps longer survival than glioma here. Okay. So we have these different categories of diagnosis, different cancer types. We might want to now make something like the Kaplan-Meier plot that summarizes the survival curve for within each of these groups. But of course, within each group, all the other features are varying, right? There's going to be men and women in every diagnosis group. There's going to be people of different age, people of different Karnofsky index. So to plot a survival curve, we have to kind of pick a typical value. Right. And so we're going to we write this little function here that's going to pick a typical value for each of the features. Having fixed that, then we could change the diagnosis. And for every different diagnosis, we'll get different survival curves. So that's what these two cells are doing. And we can let's just take a look at the plotted survival curves. So for each of the 4 diagnoses, there's a typical value for all the other variables. And this is just, the Cox model, it specifies a model for this thing we call the hazard rate, which is this instantaneous rate of failure. And from the hazard rate, you can get the survival curve. So this just plots the estimate of the survival curve for 4 different sets of covariates. The only difference between these is just what type of diagnosis. If we changed the sex or the Karnofsky index, all 4 curves would move in unison. They wouldn't look all the same, but this gives some idea about relative differences. So we see that I mentioned earlier that meningioma had a more negative coefficient, so it might be associated with longer survival. And we see that at the end of the study, 75% or 70% are survived compared to the glioma and
that HG glioma
looks very, very severe.
Looks really aggressive. Yeah.
Yeah.